Welcome back to Microlab. I hope you have been enjoying and learning one or two things from my videos, and I hope you've been enjoying my short little videos that I've been posting recently. I can see your likes, I can see your views. Thank you for subscribing, thank you for watching. So today we are going to go to gram staining. That is what we have to learn for today. So I'll just briefly explain what gram staining is before we now move to the lab work in proper, where I show you what gram staining is exactly. So what is gram staining? Gram staining is actually a fundamental technique in microbiology. We have the primary stain, we have the mordant, we have the colorization, and then we have the counter stain. For the primary stain, we have the crystal violet. For the mordant, we have the iodine. For the decolorization, we have the alcohol, and then for the counter stain, we have saffronin. So after the gram staining procedure, then that's what we differentiate between gram positive and gram negative bacteria. So the gram positive bacteria actually turns purple after you view it under the microscope, after you carry out your gram staining. Then the gram negative, they're either red or pink under the microscope. So follow me as we do the lab work. Also, don't forget to subscribe, like, and share when you get one or two things. So let's go to the lab work now. Now we are doing our gram staining. So this is a pure culture of an organism that we are going to be using for the gram staining. So we have up to 4 to 5 different organisms here that we want to do with gram staining. And then here is me lighting up the spirit lamp. So you need your spirit lamp, your inoculating loop, your immersion oil, your reagents, your 70% ethanol, your glass slide and cover slip. That is me sterilizing the inoculating loop before use. Then I am preparing the glass slides for the gram staining procedure. So that is 70% alcohol, which I'm spraying with my cotton wool to clean the cover slide and the glass slide. So if you notice that I am cleaning this glass slide, trying as much as possible to avoid the surface of the glass slide, I'm just holding the tip side, the end side of the glass slide. So I'm cleaning it with 70% alcohol and then with my cotton wool to clean the glass slide, to make the glass slide surface grease free and dust free for the gram staining procedure that I'm about to carry out. So this process is repeated for as many glass slide that you will be needing. So I take my inoculating loop back again and sterilize them. You can never sterilize your inoculating loop enough, so you have to keep sterilizing them with your spirit lamp. A Bunsen burner too can do this work, and you have to heat your inoculating loop red hot. So this is me just taking a scoop, just a small scoop of the sample. It's also called an inoculum. It's not a small scoop of the sample from the culture. And then this is me applying it to the cleaned glass slide. You have to tease the organism well on the surface of the glass slide. That's what I'm currently doing, using my inoculating loop to tease the surface of the organism and sterilizing back the inoculating loop with the spirit lamp. And you have to heat your inoculating loop red hot, allow it to cool. Then you take a scoop of the water, just a loop of drop of water from the inoculum onto the organism on the surface of the glass slide, and then you tease again. You make sure that you are teasing. This process is also you doing smear. You are smearing the organism and the sterilized water on the surface of the glass slide. So you tease it well, you smear it well, and then you place it on the rack. You are allowing it to dry. While doing that, you are also sterilizing your inoculating loop back. So allow it to cool. You take another glass slide. And you repeat the first procedure, the first process that I just finished doing. You repeat it again. See, this is me sterilizing the inoculating loop back again and again. You have to keep doing that.
allow it to cool, take a loop of water from the inoculating loop and drop it on the glass slide. You see that the first procedure is right from this procedure. The first procedure was me first taking the organism and applying the sterilized water. This second one is another method where you can first put the water before you add your organism. So this is taking another scoop of the organism. Just a scoop is enough. Taking just a scoop of the organism. So the organism is sticky. Then you apply it on the glass slide with the sterilized water and you make a proper smear. You make a proper smear of this. Can you see how I'm making the smear? You gently make the smear. Then you drop it on the rack for it to dry. So the first method is either you first take your organism on the glass slide, then you take your sterilized water on it, then you make a smear, or you first take your sterilized water on the glass slide, then you take your organism and put on the glass slide and you make a smear. So either of the two methods is just fine. So this is me doing the same procedure again for the third time, just for us to retain this procedure. You can see that I am eating the inoculating loop red hot and allowing it to cool for a while before dipping it into sterilized water to take a loop of the water. Just a loop is enough. Then you sterilize back, you sterilize your inoculating loop back, then you allow it to cool. After sterilizing it red hot, allow it to cool. I believe there should be a question why somebody should ask me why you allow your inoculating loop to cool before you scoop your organism. So you just take a scoop of the organism and then you make a smear. I'm expecting somebody to ask that question in the comment section, to ask me why do I have to cool my inoculating loop before I make a smear. Why do I have to sterilize my inoculating loop before or after I just use it to take water? You see, this is me trying to fix it. So you can either fix your smear or you allow it to air dry. But because I'm making a video, I cannot wait for that long for it to air dry. So it's fixing everything, all the glass slides. I made 5 glass slides in this video for the gram staining. So it's fixing it. Just pass it through the Bunsen burner or your spirit lamp for a while. It's not you overeating it. Then this is me clearing off my table to prepare for the proper gram staining procedure. This is me cleaning my workbench area. Always ensure that your workbench area is always clean. Then the first step, like I first said, is primary stain, which is the crystal violet. So that is the primary stain, crystal violet, that we are going to apply on the glass slide after it's fixing it. So we are applying the crystal violet on each of the glass slides that we have here. We are applying the crystal violet, which is the primary stain in gram staining, like I discussed in the introductory part of this video. So this is applying the primary stain, that is the crystal violet on the glass slides. You have to be very judicious about applying the stain. There's nothing like managing when you are doing your gram staining. Ensure that your reagent is evenly distributed. Then you wait for 1 minute before you go for the next phase. So the next phase is you rinsing your glass slide. It's not you rinsing directly on the glass slide. You have to rinse the back of the glass slide under a running water vertically, like the way it's being displayed on in this video. you have to rinse them appropriately. After 1 minute, that is 60 seconds, you have to rinse them with a running water vertically, like it is being displayed on this video. So this step is repeated 5 times just for us to get how it is like to rinse your glass slide on your running water. Then the next phase that we have to do is where we apply our mordant. Our mordant is our iodine. So we have to apply our mordant here. So that is us applying the mordant, the iodine, evenly distributing the iodine on the surface of the glass slide. So this is me showing you that is the iodine, grams iodine solution. That is what is written on that reagent bottle. You see that I am judiciously applying this reagent on the surface of the glass slide. Then I rinse under running water vertically. You try as much as possible to rinse under running water vertically. This process is repeated for the 5 glass slides that we have. Then we make use of our decolorization, which is the alcohol. Alcohol is actually the decolorization. So the difference between decolorization procedure and gram staining is that you are not going to apply it and leave it for 1 minute, but rather you apply the decolorization directly to rinse it off. So it is helping you to rinse off. You can see how I'm applying it. It's just to rinse it off. That's all. You are not applying it and leaving it for 1 minute. No, but you are using it to rinse off your glass slide. That's the purpose of the decolorizer, which is the alcohol. So we are rinsing it off. Still doing that. We are on the 4th glass slide. Now this is the 5th glass slide. So we are going to the last stage of gram staining, which is our counter stain, also known as safranin. So this is a gram safranin solution. So we apply our safranin on the cover slide, the glass slide. We apply the safranin judiciously as well, and this time around you have to wait for 1 minute before you rinse. So we are waiting. Then you rinse. After you rinse, you clean your glass slide gently with a filter paper. So this is still rinsing with safranin. And then you clear your deck. So after cleaning your glass slide with filter paper, you add immersion oil. Just a drop of the immersion oil is enough. Just 1 drop. You can see just a drop of the immersion oil is enough on your glass slide. Then you apply your cover slip on each glass slide. You gently apply your cover slip on each glass slide. That is what I'm currently doing, applying my cover slip on each glass slide. The cover slip is needed for us to view under the microscope, and the oil, immersion oil too, is needed for us to view under the microscope. There is a lot of questions I'm expecting my viewers to ask me in the comment section on this procedure that I did. Do you have any questions? Drop it in the comment section. I will always be there to give you your reply. If you have any addition, kindly drop it. We learn together also. So now I've successfully placed the cover slip on the glass slide. We are moving to the microscope room. This is where I want to view the glass slide to see, are they gram-positive or gram-negative organism. Like I said gram-positive organisms, they are purple. Gram-negative organisms are red to pink. So this is a gram-negative organism that we are seeing under the microscope. And then this is a gram-positive organism. This is so beautiful. It is purple in color. I hope we have been able to learn one or two things today. I'm expecting tons of questions. Don't forget to subscribe, like, share, and comment. Ask questions. I wish to see you soon. Have a nice day. Bye.